Okay, this is Living Environment Review. So the first section is the scientific method. So here are the parts of the scientific method. The one that kids are probably most unfamiliar with, some of the things are an inference. So it's a conclusion based on an observation or evidence, right? So you kind of know something and you're kind of trying to connect it together. Okay, so if you are giving fertilizer to a plant and the plant grows, you're making an inference that the fertilizer helped the plant grow, right? That makes sense. A theory is when you have lots of explained a natural event that's supported by strong evidence. It gets supported when you have lots of experiments that are done and this idea keeps coming out to be true. Okay? It's not a law. It's not like, we can say that it's always going to happen all the time, but it's been done enough where it's got strong support so it becomes a theory. A controlled experiment, you have to have two groups. The control group, which does not get any kind of manipulated or changed variable, and the experimental group, which gets the manipulated variable. The only way an experiment's valid is if you can compare your experimental group results versus the control group. Let's go to this idea of giving fertilizer to plants, seeing if fertilizer X helps plant growth. The only way you know if fertilizer X helps is if you have a group of plants that do not get the fertilizer. The independent variable is also the manipulated variable. It means that's the variable that's changed. The scientist decides that. If you're doing an experiment on fertilizer on plants, you decided to give that plant fertilizer. That's your variable, the independent variable. The dependent variable is what's being measured. So in this case, the height of the plants or the growth. You're likely to be given several examples where you'll have to be able to identify these different things and write about them in the Part B of the Regents. Graphing, you're definitely going to have to graph on the Regents exam. The x-axis gets the independent variable. That's the one that you changed. That's the manipulated variable. The y-axis is going to be what you're going to be measured. So make sure you have an appropriate scale. That means you have the equal amount of space between each number you put. If it goes by ones and you're going to skip a space between 1 and 2, make sure you skip a space between 2 and 3, and so on and so forth. It's got to be the same. How do you decide the scale? You have to go ahead and figure out what's your lowest value and your highest value and see how many lines you're going to need to be able to talk about all those data points. The more you can stretch it out, the better the graph's going to look. And so you want to try to use up as much space on the graph as you can. You want to make sure that if it tells you to circle your data points, you circle them. If you put an X for your data points, you put an X. Don't forget the titles of the graphs. Don't forget to include the units, things like that. The experiment can be repeated if it's a good experiment, has a large sample size. The number one way you can improve experiment is by including a larger sample size or doing more runs of the experiment. Okay? Longer periods of time, testing only one variable is a good experiment. Peer-reviewed means that scientists have looked at it and they've kind of done the same thing and they've verified the accuracy. And it's objective. No facts or opinions are mixed in your results section. Okay, that's important. The tools of biologists, graduated cylinders, make sure you always read the bottom of the meniscus. Okay, that's that curved line on the graduated cylinder, always read the bottom. Chromatography is when you have put the paper in the water, you put a dot of like some kind of ink or some kind of like chlorophyll on there, and you look to see how the colors separate out. The colors can help you identify the substance. If you have known substance and you have an unknown substance and they match during chromatography, then the unknown substance is that substance that it matched with. Balance measures mass in grams. Centrifuge can separate blood into things like plasma and red blood cells. Denser materials are on the bottom. Dichotomous keys are always on the living environment regions. So you divide things into have and have not characteristics. You have to be able to read keys. And basically at the end, it's either going to give you a direction where to go or it's going to give you a name of a group or an organism. So you can use them to identify things. Staining helps you see things easier. We stain things on slides. Oftentimes the stain is added to one end of the slide of the cover slip, the specimen being under the cover slip, and you use a piece of paper to pull or wick that stain across, and that's very helpful. Gel electrophoresis is where we take a restriction enzyme to cut up DNA. Ladies, if you want to do this, I want you paying attention to this, right? So if you can't sit together, move apart. This is what you voted for. So gel electrophoresis, when you use a restriction enzyme, you cut the DNA into fragments, and you run it on the gel, and it will sort according to size. The largest sizes will have the most difficulty traveling. They will be closest to the starting point where the wells are. The lightest or smallest fragments will be all the way at the bottom. Moves from the positive to the negative side. The microscope, we see things that are microscopic. That means you can't see them with your regular eye. Here are the parts of the microscope. An important thing to remember is the objective lenses. Usually the total magnification is the eyepiece times the objective lens, and that gives you total magnification. And so when you're viewing a specimen, you're really viewing it an increase in magnification, the total magnification size, the eyepiece times the objective lens. So for low power, it's 4x, which is the objective lens, times 10x, which is the eyepiece. So the total magnification is 40x or 40 times. Diaphragm controls the amount of light, as well as the dimmer switch. And one of the important things you want to know is that as you increase magnification, your field of view gets smaller. So it's important to always center the specimen so you can see what you're looking at. If you leave it towards the edge of the field of view, the specimen will be lost to you once you increase magnification, because you see things more magnified, but you see less of the item. It's like having a classroom view of the whole class and then focusing in on one student. It doesn't mean the whole class disappeared. They're just outside of the field of view when you focus in. Okay, characteristics of living things. Some important terms. Nutrition includes ingestion, digestion, and egestion. Okay, ingestion, taking it in, digestion, breaking it down, egestion, getting rid of waste. Excreting is another way to say getting rid of waste. Respiration is important. That's where we make cellular energy in the form of ATP. You use oxygen. You excrete carbon dioxide. You also make some water, which you will reuse for your other metabolic processes. Metabolism is an important word. Metabolic processes, those mean the chemical reactions happening within your body. Synthesis is make, when you're making things. Degradation or digestion is when you're breaking things down. We have enzymes that make things. We have enzymes that break things down. Reproduction is making more, asexual and sexual. Asexual, you're going to have the same amount of chromosomes as the individual or genes, same makeup of DNA. Sexual, you're going to get a combination of the two. So usually sex cells or gametes will have half the number of chromosomes or DNA or genes as normal body cells, and that's so that when two parents form a zygote, the sperm and the egg, the zygote then has a full set of chromosomes. Maintaining homeostasis, that means keeping a stable internal environment, is an important topic on the Regents. Okay, keeping things the same, things like blood pressure, things like temperature. You need to maintain homeostasis. Regulation refers to the controlling of homeostasis. When you see a curve for homeostasis, it will normally be like this. It will be up and down, a small little up and down curve through the graph. And in the middle of that is your set point, the line you can draw that's straight across horizontally in the middle. Those up and downs are because it's not, you can't just keep static, things are changing. Levels go up, and then you have self-regulation where you have feedback mechanisms. Sometimes they're negative feedback mechanisms, sometimes they're positive feedback mechanisms. Like a thermostat in a house, your furnace doesn't just go off, it goes on for a while, it reaches a set point, it goes off, but it might get a little warmer in the house. But then it's going to finally turn off, and then it's going to get colder. Is it going to go right on? Even if it goes right on at the set temperature, it's going to take a few minutes for it to heat back up. So that's why you get this oscillation. Blood sugars controlled this way, temperatures controlled this way, your hormones are controlled this way. Very important ways of regulation. Okay, unicellular single-celled, multicellular many-celled. This is a very important graphic talking about the levels of organization. If organelles are the smallest in the middle, they are inside cells. A group of cells that work together are tissues. A group of tissues that work together are organs. A group of organs that work together are organ systems. And finally, you have a large multicellular organism. In some shape or form, you're going to get these kinds of graphics. Parts of the cell are important. I'm not going to go over all of them, but mitochondria, that's where respiration happens. Chloroplasts, only plant cells have chloroplasts, and that's where photosynthesis takes place. Ribosomes make proteins. Protein synthesis happens with the ribosomes. Cell membrane controls what gets in and out of the cell. The nucleus controls the cell. It's the brain of the cell because it controls protein synthesis. It has the information to make those things. Active transport is when you use energy to transport things. Usually you go against a normal concentration gradient. Normally, in passive transport, you go from high concentration to low concentration. But in active transport, you're trying to move things in the opposite direction, and that requires ATP energy. Two types of passive transport you should know are diffusion and osmosis. Diffusion is the movement of substances, where osmosis is the movement of water across a semi-permeable membrane. Cell wall is another thing only found in plant cells, along with the chloroplasts and along with large vacuoles. Plant cells are usually rectangular. That's also how you can tell them from an animal cell. Animal cells have lysosomes and centrioles. Plant cells don't. Oftentimes, they'll try to get you to take body systems and compare the body systems to organelles. So for like transport, you've got cytoplasm. The circulatory system transports blood. Regulations, the nucleus and the cell membrane regulate things because the nucleus controls the cell, and the membrane controls what gets in and out. That's like the nervous system, which has brains and nerves that control your functions, but the endocrine system, which controls your hormones, right? That's why you're going through puberty and your body's changing because of the hormone levels changing, right? So two types of regulation. Respiration happens in the mitochondria where you create ATP. The respiratory system, lungs, is where you breathe in oxygen to allow it to enter the body to be able to go to the cells. You also excrete carbon dioxide. Food vacuole is like digestion. A food vacuole digests things for the cell. Your digestive system digests materials and makes them smaller so they can get to the cells. Excretory system, a waste vacuole. Your excretory system, your body is the kidneys, which gets rid of waste for all the cells. Cells are the basic unit of structure and function in all living things. All cells come from from other cells, and you need to have cells to be alive. These are the three parts of the cell theory. Biochemistry, you want to understand some major organic compounds: glucose, starch, proteins, and enzymes. Glucose is a sugar.
Anthony Davis, if you're in the building, please come up to the main office.
Which also makes it a carbohydrate. Starches are also carbohydrates, but they're more complex. They'll often show graphics of simple sugars, one simple sugar. If they show a graphic with a lot of sugars attached, that makes it a starch. Enzymes are specialized proteins. They break things down, synthesis. They make things, synthesis. They break things down, degradation or digestion. They'll show either putting things together, then you know it's a synthesis reaction. If they show things breaking apart, it's a degradation reaction. If it needs water to happen, it's hydrolysis, right? Adding water. If they talk about enzymes, they'll talk about lock and key. They'll show you a picture of an enzyme, and the substrate that it works on will have a complementary shape. If it's organic, it will contain both carbon and hydrogen, such as glucose. If it's inorganic, it doesn't have both. So like carbon dioxide is an inorganic compound. Water is the most important inorganic compound because we need that for all of our other types of activities. The 4 types of organic compounds: carbohydrates, fats and lipids, proteins, and the last one is nucleic acids. So as far as carbohydrates are concerned, simple sugars like glucose are mono. Mono means 1, monosaccharides. Disaccharides are 2, polysaccharides are many. So it's important to know they always ask you in terms of respiration, and they'll use some kind of sugar in the process to show you, and you'll have to know that it's used to make energy. Fats are lipids. So oils are also lipids. Fats are solid at room temperature. Oils are liquids at room temperature. Saturated fats, solid fat. Unsaturated fats, usually liquids like olive oil. Saturated fats, butter, Crisco shortening, margarine. And what makes them unsaturated is all their bonds are not filled. There's some double bonds in an unsaturated fat. There's not one-to-one connections with all the carbons. Proteins, long chains of amino acids, they get folded and chained into a peptide. The order and sequence of the amino acids determines the shape. Enzymes and hormones and antibodies and receptor molecules are all types of proteins. They're definitely going to show you this idea of having a complementary shape on your test. You have to understand that only things that have complementary shapes will react. Lock and key model. Remember that, well, we'll talk about enzymes down here, but enzymes can be degraded, right? They can lose their shape if they're heated too much or if they're given the wrong pH. Okay, they can break down, and then they can't carry out their reactions, which can lead to death in organisms. Nucleic acids, the last type of organic compound, A, T, C, G, and U. U is only used in RNA. So A binds with T, G binds with C. If we're talking about RNA, there is no T, so A binds with U. Back to enzymes, they're catalysts. That means they control reactions. They speed up chemical reactions. In photosynthesis, enzymes are needed. In respiration, enzymes are needed. The substrate is the substance being acted on. And the enzyme substrate complex is that lock and key when the substrate binds with the enzyme. So you can see pictures here. You have to have the exact opposite shape, complementary. Temperature affects the rate of reaction. pH affects the rate of reaction. If the enzymes break down, they become denatured, which means they change their shape. This graph is always on the Regents. It shows you that this certain enzyme works best at a pH of 3, and this enzyme works best at a pH of 8.5, 9, and that more or less they start to degrade or they're not working at optimization. The more substrates and enzymes there are, the quicker the rate of reaction will be until it levels off, until you have all of the enzymes being used at a certain rate. Dehydration synthesis is when water is removed. Hydrolysis is when you need water. So think of dehydrated, you're losing water, and hydrolysis is when you're adding water. So dehydration, water will be on the right, okay, of the yield sign, which is the arrow. On hydrolysis, water will be on the left, which is before the yield sign. Photosynthesis and respiration, hugely important. Only plants photosynthesize. All organisms, including plants, will carry out respiration. The reason plants are making their own food is so they can use it to make energy. Don't forget that. They oftentimes will try to trick you and show you graphics that will try to fool you into thinking that plants do not respirate. Do not make that mistake. Plants make food in photosynthesis. Respiration is about releasing of energy that's stored in glucose, breaking the bonds and forming ATP. Photosynthesis only takes place when the sun's out. Respiration is happening all the time. You can only live a couple of minutes without energy. You know that because you need to breathe oxygen constantly, which is also needed in respiration. If you stop breathing, you only last a couple minutes before you're dead. Chloroplast is where photosynthesis takes place, mitochondria for respiration. Here are the formulas. Don't forget the chemical formulas. Realize that they're almost exact opposites. They're reciprocal processes. The reactants of one are the products of the other. Okay? The products of one are the reactants of the other. Autotrophic nutrition is when organisms make food for themselves. Heterotrophic nutrition is when you have to go out and eat other things. Anything that's not an autotroph, okay? So if you are an autotroph, usually plants, some bacteria like blue-green bacteria, okay, and some protists. For photosynthesis, it's important that plants, the structure this mostly happens are in green leaves, and the leaves have structures that help them carry out photosynthesis. They have stomates or stomata, plural, which are the openings in the leaves. So here they're showing a particular stomate, and the size of that opening is controlled by guard cells. So plants, even though they need water, will lose water to the environment through the leaves. Water comes in through the roots, goes up to the leaves where photosynthesis takes place. The process that they get them up to the leaves is called transpiration. Plants are willing to lose some water through their leaves because they get to then move water for free without requiring energy. Think of a giant 50-foot tall tree. It has to work water and send it up against gravity. If it weren't for transpiration, which is like a siphon releasing water to the air, it would cost more energy to make that happen than the plants would get out of photosynthesis. It would be a negative yield without transpiration. So these are all related, and it's important to understand that the guard cells will control the size of the openings, and obviously plants want to control water loss, so they're not going to keep releasing a lot of water when they don't have to. However, they must always release water to keep the process flowing. Anaerobic respirations without oxygen. Yeast and bacteria do this. Fermentation is an example of anaerobic respiration. We use it to make bread and alcohol, such as wine and beer. If humans do this, it creates muscle fatigue. It creates lactic acid, which burns in your muscles and creates muscle fatigue. You did a lab on that. Don't forget that lab. Okay, rate of respiration, temperature. As temperature increases, rate of respiration increases. The amount of light, that's really for photosynthesis. The amount of water. So as you have more water available, you can increase the rates of photosynthesis. So these are rates of respiration and photosynthesis, things that affect them. The amount of carbon dioxide, as that increases, you can do more photosynthesis. The amount of minerals in the soil helps reactions happen. Human body systems, you have to understand the basics. Make sure you go over this chart. Digestion, you want to break down that food into the component organic compounds such as carbohydrates, proteins, nucleic acids, and lipids. You want to get them to your cells. The stomach has an acidic environment. The small intestine's a basic environment. Okay? Solid wastes that are not used by the body get excreted or egested are the two ways you'll see that said. Respiration, you have the mouth, you have the lungs, you have alveoli, which are little sacs that allow you to exchange gases with the blood. Remember, you are excreting carbon dioxide, you are taking in oxygen. The more you exercise, the more your rate of breathing will increase because you need to get more oxygen to make more energy. The circulatory system transports all the materials the cells need to the cells and gets the waste away from the cells. So oxygen to the cells, glucose to the cells, carbon dioxide away from the cells, metabolic waste away from the cells. Notice we just talked about all the things in respiration in terms of where they're going to and from the cells. Red blood cells carry oxygen with the help of hemoglobin, and iron is needed for that. White blood cells fight infection, platelets help clot the blood, plasma carries all the materials, that's the liquid part of blood, right? Water and other things, except oxygen. Blood pressure is a regulation of how strong the heart is beating to get blood around the body. As you exercise,
Samantha Dollard and Natalie Rubenstein, please come to the main office.
And need more
Thank
energy, you're going to need to, increase respiration. Excretory system, excreting waste, not digestive waste. Don't confuse them. Digestive waste is leftover food that was processed and not sent to the body. Okay, the nutrients we're taking out of that food sent to your cells, and now your cells did stuff with it. Respiration, photosynthesis if you're an autotroph, and other metabolic processes, growing, developing, all of those things, and now you're excreting the metabolic waste that collects in your kidneys and gets excreted through the urethra, and in your urine, water, salt, urea, and carbon dioxide doesn't get excreted from the kidneys, but it gets excreted from the lungs. Locomotion is a big term. It's your ability to move. The skeletal and the muscular system are involved in locomotion. Some single-celled organisms have things like cilia or flagella, which are structures on cells that help them move or perform locomotion. The nervous system helps you sense the environment and regulate. Nerve cells, they always have pictures of nerve cells, and they show these little gaps. It's called the synapse between two different cells. And the synapse, they'll often show chemicals being released, and those chemicals are attaching to receptors. Again, another example of lock and key model. Endocrine system is how we regulate our hormones, help regulate bodily functions. Glands secrete hormones. Hormones, again, have specific shapes that attach to receptors. The menstrual cycle is a big one, talked about on the Regents. It involves the release of the eggs from the ovaries and causes different changes in the uterus that allows the ability for an animal, and humans are animals, so to carry an embryo. Okay? Estrogen and progesterone are the hormones that get sent from the ovaries to the uterus that cause the lining of the uterus to be thickened, and eventually, if it's not used, then other hormones will cause the release of these things. If there's not a fertilized egg, and that's menstruation. Females, that happens once a month. Some other hormones, pituitary gland, which is the master gland in the brain, releases the TSH and FSH. Thyroid stimulating hormone, which regulates your metabolism. Follicle stimulating hormone, and LH. These are sent from the ovaries and are involved in the menstrual cycle. Follicle stimulating hormone will allow a follicle or an egg to be released into the uterus from the ovaries, and LH will be used in conjunction to help, shed the increased thickening of the lining of the uterus if it's not used. Thyroxine from the thyroid gland Adrenal gland releases adrenaline. Pancreas, insulin and glucagon. Insulin lowers your blood sugar level in the body. Glucagon increases your blood sugar level in the body. So people that have diabetes do not have enough insulin, and so they have a hard time regulating their blood sugar levels. Estrogen and progesterone for the menstrual cycle and reproductive hormones, mostly in females. The testes produce testosterone, which causes male secondary sexual characteristics. Immune system, you're a pathogen, you are the invader. Microbes are pathogens, viruses are pathogens, some fungi, some bacteria, some protists that are parasites. Antigens are proteins that are found on the pathogens, and they are what causes an immune response. Your body recognizes these foreign proteins and will send antibodies to attach to the antigens, and if that happens, the white blood cells go and destroy it. So we use things like vaccines to stimulate an immune response to create antibodies, things that will latch onto antigens should the invader, the pathogen, ever come in the body for real. Antibodies stay with people for a time period, depending on the disease, some more than others. So if the disease comes back, you'll be able to fight off the infection before it makes you sick. Vaccines are a dead or weakened virus. We have new vaccines which are made using RNA and protein synthesis. Antibiotics are drugs that are helpful in fighting infection against bacteria, not against viruses. There's a problem with bacteria building resistance to our antibiotics today because we keep giving antibiotics to people unnecessarily. Because people go to the doctor and they're sick and they want to get something out of the visit, and they don't like to hear that it's a virus and there's nothing that can be done but rest. And so they'll often get prescribed antibiotics unnecessarily, which does nothing except help bacteria get stronger and stop immune responses in the future for people that need it. Allergic reactions, when you respond to something, you have an immune response, but the thing that you're responding to is not harmful. So in that case, your body will produce histamines as part of its immune response, which causes swelling and itching and breathing difficulties like asthma. So that's why people get those symptoms, but they don't get sick from asthma. Here's a nice picture of the antigens, which are the proteins on the pathogen. Here are the antibodies. Notice the complementary shapes to attach. And the antibodies become signals and say, hey, white blood cells, come and destroy this cell. It doesn't belong here. Okay, so malfunctions of the digestive system are listed here. You can look at those. They're good to know. You don't have to memorize all the stuff I'm talking to you about, but you should be familiar with it enough so that when you see it on the test, you'll know what to do with it. Okay, so definitely review this worksheet. I'm not going to go over these. You can read these. Some we've already talked about. I will talk about AIDS. AIDS is called autoimmune deficiency syndrome. It's when a virus attacks and destroys the white blood cells. And so what happens is you can't fight other infections. So you can die of things like pneumonia. So someone can have HIV, which is the virus, but not have AIDS, which means, your white blood cells are keeping at an acceptable level. Feedback mechanisms, the ways that we can control the regulation and homeostasis. So when it's cold outside, you shiver. That helps warm your body up to bring it back to 98.6 degrees. When it's hot outside, you sweat, bring your body back to 98.6 degrees. When your sugar levels get too high, the pancreas secretes insulin that lowers your blood sugar level. When your blood sugar level gets too low, the liver releases glucagon, which helps to then release sugar back into the blood, raising your sugar levels. Here's this idea, that graph I was telling you about homeostasis. If these are the levels and this is time, you can see that over time we get this up and down effect. Homeostasis would be a line right through the middle here, because things do alter slightly on a given day or a given time or a given moment. Okay, asexual, one parent, identical offspring, sexual, two parents. Each parent gives half the genetic information. Mitosis is involved in cell division. Mitosis is actually when you take the genetic information, you double it, and you divide it into two new cells. However, it's often associated with cell division because you can't have cell division without mitosis happening. Meiosis is the creation of sex cells. The daughter cells will have half the genetic information of the original parent cell. These cells are considered gametes, and they're used in sexual reproduction so that the zygote, which forms from fertilization, will have a complete set. Sometimes you'll see asexual reproduction in different graphic forms. Here's a hydra, which is a protist that buds. Here's probably an amoeba, because I see pseudopods here. It goes from one cell and just divides into two. And here's a yeast cell, which also is budding, all forms of asexual reproduction. It's faster, but it does have no variation. So if there's some type of sickness or pathogen that affects the population and kills one, it'll probably kill all of them. Where in types of ways we can talk about asexual reproduction, binary fission, budding, regeneration, vegetative propagation. Those are all ways of cellular asexual reproduction, and it's all mitotic cell division, which is mitosis. Here's the diagram of the stages of mitosis. It's important to realize that we have to divide up the chromosomes, and that's what these stages are here. To get two new nuclei for two new cells that are about to finish dividing. So if you have 8 chromosomes in a parent, you'll have 8 in each of the offspring. Sexual reproduction, the advantage is you have variety. Every organism is a little different. So if a pathogen is going to kill an organism, it's not going to kill all of them. Disadvantage is you have to find a mate. It takes a lot of time. In meiosis, you start out with 46 cells. You go through a mitosis 1, they call that, where you're going to end up with 46. So this is just like mitosis right now. But then these cells divide again without doubling, and you end up with 4 haploid cells, 4 gametes that have half the number of chromosomes. The homologous chromosomes separate and go into 2 of the cells. Since we have it twice, that's why we get 4 cells. Another way you can get more variation is by crossing over. That means these homologous chromosomes, before they separate into the 4 cells, they'll exchange certain genes. So those dominant recessive genes, the different forms of them, they will cross over and sometimes swap places so that we can get even more genetic variation in those gametes or sex cells. Fertilization is when the gametes come together. So a zygote will have a normal number of chromosomes. So if you start out with 2N, for humans it's 2N or 46 chromosomes. If you're only N, you're 23 chromosomes, and that's a gamete, half the number. You have to be 2N because each is a pair. That means 2 of these. 2 of these, 2N, versus 1N is having only one of those, this one or this one, this one or this one, this one or this one, so on and so forth, for all 23 pairs. When an organism divides, as soon as it forms a zygote, it's a single cell. It will start to go through cleavage, which means a lot of mitosis, a blastula, a gastrula. They just keep dividing over and over again, and eventually you're going to get differentiation. That's when you use different parts of the DNA, of the genetic information, turned on in different cells to make different cell types. Nerve cells versus body cells versus other types. Stem cells are undifferentiated. They can turn into any type of cell. Placenta is the connection point between the mother and the fetus. The blood of the mother and the fetus never mix. That's important. The mother and the child, the fetus, will exchange nutrients, gases, and waste, but their blood never gets together. So if you take drugs or alcohol, if you use them as a mother, it will definitely go into the fetus. Smoking, bad diet, all of this stuff affects the fetus. It can cause birth defects, low birth weight, and even deformities, right? Mutations in the cells. Males have testosterone, females have estrogen and progesterone. Flowering plants, sometimes they'll talk about flowering plants. The stamen produces sperm, that's the male reproductive structure. The pistils, the female, produces the egg. You still get a zygote formed. So first you have to have pollination, getting the pollen from the male structure to the female structure. And then you can have fertilization, which is the sperm and the egg fusing. And then that forms a zygote, which is captured in the seed. And that's how reproduction occurs in plants. Okay, you can go over these on your own, but this is an example of differentiation, going from an embryo to forming those different types of specialized cells. This is the fertilized zygote. We can see cell division, cleavage, bastula, gastrula, but eventually we're going to start to get to an embryo and then to differentiation. This is an example of fertilization. This is showing N number of chromosomes. These are 2N, 2N, 2N. Genetics, a study of heredity, parents passing down traits from parents to offspring. Homologous chromosomes are the pair of chromosomes. We saw one go into each of the gametes during meiosis. Genes determine your traits, which are your inherited characteristics. The order of organization: cell, nucleus is inside the cell, chromosomes are carried within the nucleus, and DNA makes up chromosomes. If they threw genes in here, genes would be after chromosomes because it's smaller, and again makes up DNA. Chromosomes are made of DNA, which is a double-stranded twisted ladder. Don't forget how they bond: A with T, C with G. If you're RNA, there is no T, it's A with U. DNA replication, you have to know this, is when we take a double strand, we split it in half, and we can make copies of that by bringing in other nucleotide sequences because we know the way they're supposed to match. So these two strands are identical to the original strand. And this is happening in mitosis. This is how mitosis actually happens, how we double the sets of chromosomes. Protein synthesis happens at the ribosomes. The DNA message is made. We make a copy of that messenger RNA after the DNA unzips. So the messenger RNA gets sent to the nucleus, out of the nucleus into the cytoplasm, gets picked up by a ribosome and read. An additional RNA called transfer RNA brings in amino acids, which it captures from the food you eat that's floating around the cytoplasm, and brings it into the correct place based on a 3-base sequence called a codon. So the messenger RNA will have the codon, and the transfer RNA will have the 3-sequence anticodon. They have to match and join together. Sometimes we have mistakes in making proteins. Mutations can cause cancer. It's uncontrolled mitosis. Radiation can cause that. Chemicals can cause these changes. They will only get passed down if they happen in the gametes, if it happens during meiosis. Types of mutations: addition, we add a base; deletion, we delete a base; and insertion, we insert a base. We can also have substitution, which is a base for a base. Some things are worse than others depending on how much they change the sequence. Addition, deletions, and insertions are very bad because you change the whole sequence after it, especially if it happens at the beginning of the message. Whereas substitution, 1 for 1, only affects one amino acid different, so it's not that bad. We can try to get desired traits by selective breeding. That means we take animals and we breed them together because they have traits we like in the hopes that they will produce offspring with those like traits. Racehorses, cattle, vegetables, we use all of these ways to get the desired outcomes. Biotechnology, we can actually change DNA inside organisms. This is a way that insulin is made today. We can take a section of DNA, like in a human, like an insulin gene. We can insert it in little circular plasmids of like a bacterial cell, which is a little circular section. And now we can get, so we can use a restriction enzyme to cut that plasmid. It now has an opening, and we can insert the human gene in there. And now we have a new plasmid with a human gene, and we insert that in bacteria. And now every time the bacteria reads that, it's going to read the human DNA and make that product, in this case, insulin. Cloning, a way of making an identical copy of an organism. Gel electrophoresis is where we are breaking up DNA. We use that to compare and contrast. We use that in crime scenes to try to track down and find individuals. We use that in seeing which organisms are most similar if we're looking at evolution. Ecology, levels of organization. Species is the individual. Population is a group of the same species. Community are different species living together. Ecosystems include the biotic and abiotic factors, that's the living and non-living factors such as air and water, and the biosphere is the earth. An ecosystem is when you have organisms living together, and they are, again, are affected by the abiotic factors as well. The sun is the ultimate source of energy for any ecosystem. Okay, photosynthesis, we're taking that sunlight and putting it into the organisms in the form of stored food. Decomposers break down dead things and recycle nutrients into the soil so plants can use that to grow. And there's got to be a flow of energy. It's got to be a cycle, right? Energy's got to return back to the earth. Whenever you add a to a term, it means non, so biotic is living, abiotic is non-living. Autotrophs make their own food, and are producers. Heterotrophs do not make their own food. That means they have to ingest or eat other things. They are consumers. Herbivores eat plants, carnivores eat meat, omnivores eat both. Carnivores have predator-prey relationships, which are a hawk and a mouse. Decomposers break things down. Fungi and bacteria, they return nutrients to the soil. Some organisms have different relationships, like a parasitic relationship, like a tick and a human or a tick and a dog. It's one gets harmed, the other one gets helped. So the parasite helps itself but harms its host. There are other types of symbiotic relationships, such as mutualism, where they both help each other, and commensalism, where one's helped, the other's not, like a barnacle and a whale. Barnacle gets help with food and housing. The whale's not helped, but it's not hurt. Carrying capacity, like homeostasis, is kind of like the homeostasis of the environment. You'll often see a squiggly line showing populations going up and down slightly. The carrying capacity is the line in the middle. What controls the carrying capacity? The amount of food, water, living space, the availability of mates, things like that. If you disrupt an ecosystem, you decrease biodiversity. Severe storms like hurricanes, floods, droughts do that. Food chain, food web. Food chains, direct relationship. Wheat, grasshopper, to frog, to snake. Food web, interrelationships. An energy pyramid will show the relationship of energy as it goes up the pyramid. Energy is less, decreases. That's because animals and organisms metabolize, and a lot of the energy is lost to the environment as heat. These arrows show energy relationships. The wheat does not eat the grasshopper. The energy from the wheat, when the grasshopper eats it, flows into the grasshopper. Very important. That's always on the regions. Ecological succession. One community replaces another. For instance, we have a primary succession. That means first. Primary means first. So you have like a volcanic eruption. You have bare rock. And eventually you'll get things like lichens, and you'll get they'll make some soil, and then you get grasses, shrubs, trees, and obviously animals will start to come. When you get grasses, you'll get flowers, and then bees will come, and all sorts of animals and things, right? Eventually you get a forest. A climax community is when the ecosystem is very stable, the most biodiversity. If a forest gets destroyed or a field gets destroyed from farming or like a fire, this is called secondary succession. It doesn't really, nothing gets completely burned out, there's soil at least, and so things will progress similarly, but you don't always have that lichen stage, which is that algae and a fungus that can create soil by breaking down rock and things. Biodiversity is good. You have a lot of organisms like the rainforest. Human impact, depletion of resources. Okay, we're using up too much stuff. Okay, we're using faster than we can renew them. Examples, things like non-renewable resources like coal, oil, natural gas, finite resources like food, clothing, shelter. Overhunting, overfishing decreases biodiversity. We can stop that by having conservation programs that protect species. Importing species is a bad thing. That's when you introduce invasive species to an environment. They don't have predators and natural enemies, and so they proliferate and crowd out and use up the resources for the other organisms that are natural to the area. Overgrazing and overcropping, or when you use up too many resources, you don't rotate the crops, and so you deplete the soil and can't grow things after a while. Deforestation is when you deforest an area like the rainforest, very bad. You can't get things like that back. Direct harvesting is when you remove one species, which can go ahead and lead the extinction of the species. It can also upset the food chains and food webs. Acid rain, when you are creating too much acidity in the water caused by pollution from factories, a lot of sulfur dioxide and carbon dioxide. Global warming is when we are making the earth warmer. We're creating climate change, which leads to global warming by burning of fossil fuels and releasing extra carbon dioxide to the atmosphere. Deforestation also adds to that because there are less trees to soak up the carbon dioxide. Ozone depletion has caused holes in the ozone layer, which allows us to be exposed to more ultraviolet radiation. It's harmful for plants. It's harmful for humans, can cause cancer. It's caused by CFCs. It is a bit of a success story. The hole is getting less because we've stopped using really bad chlorofluorocarbons, which were in a lot of products like Freons and hairsprays and aerosols. Industrialization is good for the economy, but creates a lot of pollution, takes away environments. Burning of fossil fuels leading to pollution, leading to global warming. There are ways you can combat that by not eliminating it, but you can use public transportation. You can try to reduce your energy usage. Waste. Problem with waste is when we dump things when we shouldn't, when we over-fertilize, when we use pesticides on our lawn to control pests and things, because this pollutes our water. When you dump waste directly into water, reduces biodiversity, creates mutations in animals and plants. Spraying of pesticides, we kind of included with that. And that's pretty much the review. So good luck for your Living Environment Regents.